Welcome to the Breaking Down Biology blog Q&A session. My name is Neha Lozano, and today I am joined by Dr. Stephanie Gaw, senior author of today's article, as well as Dr. Nida Ozarslan, the lead author of the article we will be discussing today. Placental malaria induces a unique methylation profile associated with fetal growth restriction. It is great to have both of you here today.
Thanks for having us.
Could you elaborate more on the significance of the BMP4 gene, such as how its location and traits altered your understanding of non-malarial FGR and placental malaria FGR? Why was this discovery especially significant?
First of all, when you think of DNA methylation, you can think more like a light switch. When you methylate the DNA, it's dimming the lights in terms of the gene is not expressed as high. When you unmethylate it, when it's hypomethylated, you're increasing the light, so you see that gene more transcribed. With BMP4, what we saw was it was commonly hypomethylated with both placental malarial FGR and a non-malarial FGR. So we wanted to look at RNA expression, which we would expect increased expression level of the RNA. However, interestingly, we didn't saw the differences on an RNA level. And this could be explained by a coping mechanism that the placenta is trying to regulate the changes that can occur with both malarial and non-malarial FGR to get them back to the same level.
I think the interesting thing about BMP4 was that this was the only one that overlapped between the two. It was very, very significant also. So that implies that actually this gene is really important in regulating fetal growth, no matter what kind of insults happen to try to affect how the baby grows.
How exactly did you screen all the methylation profiles of the samples when you compared them?
So we started by extracting DNA from placental biopsies that we collected right after delivery. And then we used a platform called Illumina Infinium Methylepic Array that looks at over 850,000 CpG sites. And again, CpG sites are little light switches that you can think of in terms of regulating the transcription of the genes. Out of those 850,000 CpG sites, we did quality assessment to make sure all the data that we're looking at is reliable. And we ended up with over 780,000 CpG sites to analyze and look further into it. The most striking results were when we looked at malarial FGR versus non-malarial FGR. Overall, when we say hyper and hypomethylation, it was always relative when you compared the two groups. That's how we identified the significant differentially methylated CpG sites and looked at which genes they were related to, their location, compared to the genes, because not all CpG sites affect the transcription of the gene. It has to be on the promoter region, the region before the gene, to affect its downstream regulation. Also, we looked at the ones that were in the promoter regions and kind of tried to understand the function of those genes, like how does methylation affect the gene transcription, and that gene transcription affect the functionality of the cell.
Thank you. Okay, my very last question is, what new findings occurred as a result of the study?
So we can say that non-malarial FGR and placental malaria-related FGR are very different in terms of their epigenetic profiles, like methylation levels. And this kind of makes sense because we know that placental malaria is an inflammatory environment where the placenta encounters the parasite and infected red blood cells. What was also special was that in our placental malaria group, we saw some changes that could be linked to neurodevelopmental outcomes through the methylation changes. And in clinical studies, this has been already identified where infants who were born to mothers that had malaria in pregnancy can have worse neurodevelopmental outcomes. So that was really striking for us to also see this on an epigenetic profile level. Overall, our findings don't immediately translate to treatments because like Stephanie said, this is a preliminary study looking at the epigenetic profiles for the first time. But this kind of opens a door for follow-up work for larger studies, and also looking at different types of epigenetic regulations, because the DNA methylation is just one type of it. There are other histomodifications that could be investigated downstream. We also could use this to identify potentially a downstream biomarker that can help identifying these pregnancies that are at higher risk for FGR for like the downstream like treatment intervention.
Just like Nida said, the best studies produce more questions rather than answers. This is really interesting. This is not what we expected at all to find. And so, the questions that I have moving forward are, how important is BMP1? Why is that gene so tightly regulated between the two types? What are other ways that these genes can be regulated? Because it may not be through methylation, as Nida alluded to. It might be through histone modifications or non-coding RNAs or other ways that gene expression is moderated between different conditions. So it might be something else. And hopefully this will help us understand, how these impacts that happen to the baby in utero will affect their long-term outcomes.
Great. Thank you both so much. It's really lovely to have you guys, and your research is incredibly inspiring. And I'm really excited for the blog's release.
Thank you so much.